Hello students, I'm Anusha Suri, I'm your biochemistry faculty. In this section, we will be learning about immunochemistry. Immunochemistry, in the sense here, we will be learning about immunoglobulin, structure of immunoglobulin, structure of your antibodies, immunoglobulin otherwise your antibodies and their properties. And we will be learning about the various diagnostic tests, that is about the ELISA. Now, starting with your immunoglobulin structure. So before we go into that immunoglobulin structure, we will have a look on what is antibodies and what are antigens. So we have a wonderful immune defense system in our body. Our first line of defense, it is, we get right from the birth, we are, we have well developed immune system in our body. Immune system is nothing but what the resistance which a body exhibits towards the infectious diseases, we will call it as the immune system, and that procedure or the process you will call it as the immunity, right? So your immune system, the main components of your immune systems, that is your antigens as well as the antibody. Antigen is a foreign substance. It can be virus, it can be bacteria, or it can be any other particle which is entering into our body which is not as foreign. That is nothing but your antigen. Antibodies, they are the glycoproteins which are produced by the vertebrates in response to the antigens, is nothing but your antibody. Rodney Porter and Gerard Edelman in the year 1959, they have elucidated the structure of your antibody. How the first structure of the antibody has been explained by them. And here, before we go into the structure, how does this antigen and antibody how do they interact? We'll just have a primary glance on it. So your antigens, they in response to the antigens in our body, various antibodies are being secreted out, they are produced out. So your antigen as well as the antibody, they have the specific interactions and non-specific interactions. Specifically, your antigen and their antibody, they will go and bind non-covalently with each other, and once the reaction is facilitated, then they are separated out. So usually the antigen antibody interactions they have various different mechanisms. It can be, precipitation, agglutination, diffusion. So like this immunodiffusion, fluorescence methods, like this via the various mechanisms your antigen and your antibody will interact, and then those interactions, they're very much responsible for the immune system of our body. Now we will see in detail about the structure of the antibody. See, structure of your antibody. Antibody, it is a Y-shaped molecule. This Y-shaped molecule, it contains two light chains and two heavy chains. So two light chains which has a molecular weight of 25,000 Daltons, whereas your heavy chains, they have the molecular weight of 50,000 Daltons. Now each light chain, the number of amino acids present in your light chains, it is your 214 amino acids are present in your light chain, whereas 450 amino acids are present in your heavy chain. Each light chain is linked to your heavy chain, and both of your light chains are linked to each other via a Y-shaped structure. That is why you will call this structure as your Y-shaped structure, and this is a globulin. They are nothing, your antibodies are nothing but your protein molecules, are the glycoprotein molecules. So this is exhibiting the structure of your globulin, and this structure we will see in detail. How does this Y shape it is and what are the other components that are present in it, we will see now in detail. Now, if you see, this is the structure of your immunoglobulin. This is your light chain, and this is one is VH heavy chain, and this is your light chain. The one which is depicted in the blue color, which is your light chain. This is your light chain, and this is your heavy chain. Right now, your light chain and heavy chain, they contain the special regions. Those first region you will call them as a variable regions. So variable regions, that means they are varying. So those variable regions, the first they contain 110 amino acids. First 110 amino acids near the N-terminal region. This is the N-terminal region. This is your C-terminal region. Near your N-terminal region, that is your amino region, both your heavy chain as well as the light chain, they contain highly variable sequences, variable amino acids present here. The first 110 amino acids, they are highly variable, and these amino, because with respect to the heavy chain and light chain, they are represented as V variable heavy chain, variable light chain. Coming to the next region, that is, this is your constant region. So if you see, this is your constant region. In the constant region, that is from the region 110. So your 110 onwards, 110 to your 450 amino acids, if they are present, that is your heavy chain. Whereas 110 to 214 amino acids, they are present, that is your light chain. So your light chain contains your 110 to your 204 amino acids. So you will denote it as your Cl. This you will denote it as CH. So your CH region, that is your heavy chain, it contains your complete from 110 onwards 450 amino acids. Whereas your light chain, it contains from 110 onwards the remaining 214 amino acids are present in your light chain region. Now if you say this is your antigen binding site onto this region, your antigens are being bound. Now this structure you will call it as your immunoglobulin domain. That means where both your light chain as well as your heavy chain, they are having the homologous units. Homologous units means same variable units they are present. And first 110 amino acid region, it is so same, they have same amino acid residues, they are there. So they will form a globular motif like structure that you will call it as your immunoglobular domain or immunoglobulin domain. Now with each domain, so there is one intra-chain disulfide bridge is present here, and each domain, it contains a loop of almost 60 to 70 amino acids are present here. And then the next, if you see each domain of your heavy chain, in coming to your variable region, they contain 3 to 4 constant regions. This is your 3 to 4 constant regions are present in it. Next, coming to this part, this is your hinge region which you are seeing. Before, see, this is the hinge region of your, which is trying to connect. We are getting a Y-shaped structure here. So before that, we should see what is this here, hypervariable regions. So variable regions, if you see here, these are your variable regions, whereas hypervariable region, you are seeing these 3 different lines here, 3 different variations you are seeing. What is this hypervariable region? Hypervariable regions are nothing but those regions wherein your small areas of which are present both in your light chain as well as in your heavy chain, they are present. They are present in your light chain, they are present in your heavy chain. But the difference is that there is a variability among these chains. So why is this variability? They are varying with varying from one antibody to the another. This, because of this variations only, we get various classes of your immunoglobulins because of this variable regions. And onto this variable regions only your antigens will go and bind. These hypervariable regions, they are very much specific and they are complementary to the antigen, and that is why those regions we will call them as your CDRs, that is your complementarity determining regions. Thus we will see now in detail. We are talking about complementary determining regions, that is your CDRs, we call them as. These regions which are marked that they are your CDR regions. Now this is your whole immunoglobulin structure, that is what we have learned. So this is your CL region, light chain region, CL light. This is your VL, that is your variable light chain region, and this is your variable heavy region, right? So if you see here, you have your CH constant heavy region here, heavy chain region, and here CH1, CH2, CH3, they are present. Of CH is a constant, H stands for your heavy chain. So your constant regions for your heavy chain 1, heavy 2, heavy 3. So these are what, these are the various regions which is your heavy chain on heavy chain regions which are showing the same number of your amino acids, same. I mean like they're showing your amino acid sequences from your after your 110 onwards to your 450 amino acids, right? So here if you see, this is your CDR1, CDR2, CDR3 regions. So these are your complementarity determining regions. So what is this complementarity determining regions? They are the specific regions onto which your antigens will specifically come and bind on this particular regions. So here you have framework regions also. Framework regions means a relatively an invariant sequence of peptides which are present in your variable region of your antibodies, of your light as well as your heavy chains, you will see them. So these variable regions which almost constitute the 70% of your total variable region, Those regions, they are acting like a scaffolds for your CDRs. Like means they're supporting your CDRs. Those regions you will call them as your framework regions. Now, if you see in detail here, the region between your CH1 and CH2 domain, this domain we will call it as the hinge region. This is like a hinge which is holding your above two light chains and your two heavy chains it is holding. So, this region you will call it as the hinge region. So, this is the short hinge region which is made up of your 10 amino acids. Those 10 amino acids, they are giving rise to a chains called as your alpha chain and your gamma chain like structure, whereas your long hinge region, it will consist of your 60 amino acids. Out of the 60 amino acids, so usually it will give you your gamma as well as your delta chains, and the 60 amino acids most commonly that you will see in your hinge region are your proline, your cysteine, lysine, aspartic acid. These are observed, these kind of amino acids you will usually see in this hinge region. Because of the presence of this open structure, it is making susceptible for the proteolytic cleavage. If you see, there are the presence of your disulfide bridges also. So this is your heavy chain, this is your light chain, and this is your variable regions onto your heavy chain as well as your light chain VL. This is your VH. So like this, in this way, you have your whole structure of your immunoglobulin. Now, other than Y-shaped chain, we have the J chain structure. That is also your glycoprotein. It also, gives your two or more units combined together, making a polymeric structure. But the important part of your J proteins is that it is, J structure, J chain structure. It has a secretory component into it. So, a secretory chain that is joining and giving rise to your J-shaped structure. This secretory component, it is very much helpful because of presence of this secretory component. This particular J chain shaped structures, the immunoglobulin structures, they play a very important role in the secretory activities, like for example secretion of saliva, secretion of tears, secretion of milk from the mammary glands. So like this secretory component role, it is being played by this particularly immunoglobulin because of this secretory component being present. And here the best example is your immunoglobulin A, which acts like a major secretory immunoglobulin. Now, along with the secretory component, your J chain is attached with the disulfide bridges, that is making your carboxyl terminal, and then you're getting your N-terminal end. And your secretory component that has been formed, this is secretory component, it is formed because of the proteolytic cleavage. Because of the proteolytic cleavage of the secretory component, you see it in the different fractions, and entirely it is joining the J-shaped globulin structure. So we can call it as a product of your proteolytic cleavage. The secretory component is a product of the proteolytic cleavage. Now we will be learning about classification of immunoglobulins. Now we will see, based on your structurally, how your immunoglobulins are classified based on the structural differences as well as the functional differences. How they are classified, we will see now. So they are classified into 5 different classes: immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, immunoglobulin E. So into the 5 different classes, your immunoglobulins are being classified based on their structural arrangements, based on the presence of the polypeptide chains that is present, whether it is a monomeric or whether it is your dimeric or whether it is pentameric, and based on the functions. So now we will see each and every function of it. Now, immunoglobulin G, it is 80% it is present in your serum, and it plays a very, very important role in our body. It is a monomeric structure. It contains 2 light chains as well as 2 heavy chains. And coming to the classification, it has been classified further into 4 different types. That is your immunoglobulin G1, G2, G3, and G4. Coming to the functions, it is like it crosses the placenta. This is the one immunoglobulin that is crossing the placenta. It plays a very important role in the opsonization process of our body. And your G1, G3, and G4 classes, they play an important role as the complement system activators, as well as they play an important role as the opsonins. So they have their heavy chain as heavy chain and light chain between the K and as well as the lambda. And coming to your G, it is represented as your globulin G part. And coming to the molecular weight, it has 1,50,000 Daltons in its molecular weight. Now the next class that we are seeing is your immunoglobulin A. A, it is a, as we have learned earlier, it acts like a secretory component. It is showing the J-shaped structure. It is monomeric in nature, that is like, but it however it is dimers also, like each monomer it contains two light chains, two heavy chains, and then most importantly it constitutes 10 to 15% of the serum or the plasma. And coming to the most important functions of this particular immunoglobulin, it plays a very important role in the secretory functions like secretions of your tears, secretions of your saliva, secretions of the milk from the mammary glands, and also it plays a very important role in the secretions of the digestive tract and the digestive system. So it has a molecular weight of 1,60,000 Daltons, and it does not have any further classifications, but its heavy chain it is representing your A. So that is your A alpha part it has been representing. So the next class that we are seeing is your IgM. IgM it constitutes 5 to 10% in the serum. Coming to the structure, it is your pentameric structure. That means each monomer they combine together and they give rise to this pentameric structure, five ring structures completely. It's a ring structure. It is having five different monomers, each monomer containing two light chains and two heavy chains. Coming to the important functions of this particular class, so it is a first synthesized immunoglobulin, and it plays a very important role in the immune response of the body. And here it plays a very important role in initiating or in enhancing the agglutination process, but it will not facilitate the opsonization process. No opsonization is seen with respect to this particular class immunoglobulin M. So it has a molecular weight of 900,000 Daltons. Now the next class that we are learning is your immunoglobulin D. It constitutes almost 0.2% of the serum concentration of the plasma concentration. It is a monomeric in structure. It contains two light chains as well as two heavy chains. And coming to the molecular weight, it has almost 1,80,000 Daltons in its molecular weight. And in functions, so it plays a very important role in the lymphocyte activation. It plays a very important, like it tries to be on the surface of your B cell lymphocytes, thereby B cells, thereby enhancing the B cell lymphocyte response. And it is represented heavy chain, it is represented in the form of your delta, and that is D, IgD, that's why we call it class delta. And coming to your light chain, it is represented as K or lambda. So this is about your immunoglobulin D. The next class that we are learning is immunoglobulin E. Immunoglobulin E, it constitutes almost 0.002% in our total serum concentration. And coming to it, it is a monomeric structure. It contains two light chains, two heavy chains, and molecular weight, it constitutes almost 1,90,000 Daltons in its molecular weight. Coming to the functions, it plays a very important functions in the hypersensitivity reactions as well as the allergic reactions of the body. And coming to your light chain and heavy chain classification, you see here your light chain is always, it can be your kappa particles or the lambda particles. And coming to your heavy chain, it represents in the form of E, your epsilon. It is represented in the form of your E. So that is by IgE. So this is all about your immunoglobulin E. So we have seen five different classes of your immunoglobulins and their functions, their structure.